Hello and welcome everyone to our Meet the Drug Developer webinar series. So today we are very fortunate to hear from Arthex Biotech about their progress and their path towards clinical development. As usual, a little bit about the Myotonic Dystrophy Foundation. Our vision is, of course, that we see a world with treatments and a cure for myotonic dystrophy. The mission of the Myotonic Dystrophy Foundation is community care and a cure. So we support and connect the myotonic dystrophy community. We provide resources and advocate for care, and we accelerate research towards treatments and a cure. As many of you know, we provide a wealth of resources and support. All of this information is located on our website. So there you can find information about toolkits and publications. You find information about our support programs. Our website hosts the calendar of events, and our website also hosts the digital academy with hours of scientific and curated content. And that's also the site where you will see, hopefully by sometime next week, a recording of today's webinar. We have announced it before. We are very excited about our annual conference next year in May in Indianapolis. Registration is open for community members and DM professionals, so please be sure to check out information about the hotel with a discounted hotel room block, community sessions, and a new Sunday morning workshops. Again, this is all on our website. Also, as many of you know, we are hosting these webinars on a regular basis, and we have several programs lined up for the end of this year, as well as for 2025. And again, the information is on our website, and recordings are also on our website. So now we go through the scientific content of this presentation, and here you can see a depiction of the DM drug development pipeline. And to me, that is the most important indicator of progress and hope. So not only do we have more than 20 programs, so commercial programs, we are not even listing academic programs in clinical development with known and publicly disclosed development programs, but 9 of them are actually in various phases of clinical testing. So you can find this information again on our website, and here is an updated version that was updated in September of this year. So when we think about clinical trial activity, we can stratify them in 4 broad areas, and these areas they essentially follow the central dogma of biology. So from left to right, we start with the DNA, with the gene, with a faulty repeat expansion. The gene that gets converted makes it into the RNA through a process called transcription. Because of the expanded repeats, the RNA then misfolds and forms these highly ordered, highly structured hairpin formations, and they in turn act as trap, and they trap and recruit important cellular proteins. If these proteins are not available, the end effect is not normal cellular function. So in this first category of activity, we see inhibition of transcription and gene editing. And while in the past we have not seen any activity in this space, this is slowly going to change. The second category focuses on the inactivation of toxic RNA. So here the interventions are to get rid of these toxic structures and to then free the proteins. The third mechanism is to compensate for trapped proteins, and the last mechanism is to broadly compensate for widespread cellular dysfunction. So today we are hearing from Arthex Biotech, and they are actually active in this third silo. Next one. So we are very fortunate to hear today from two folks from Arthex. So we are going to hear from Beatriz Llamusi. She's a chief scientific officer and co-founder. So she holds a master's and a PhD in biochemistry from the University of Miguel Hernández. And her domain expertise are rare neuromuscular diseases, mechanisms of diseases, and approaches to treatment. She's also the co-founder of the company, and the company was founded in 2019. She was the CEO until 2023, and she chaperoned the company from spin-off phase to raising Series B. In her role as the chief scientific officer, she's responsible for all discovery, research, and non-clinical activity. And then we are going to hear from Judy Walker. She's the chief medical officer. So Dr. Walker is a clinical neurologist by training, and she has deep domain expertise in clinical trial investigations in Canada. She also spent the last 25 years or so in a variety of clinical and medical leadership roles in the US, in Europe, and in Asia. At Arthex, she's responsible for all clinical and medical activities, so moving their lead compound through the clinical study phases, preparing for ongoing development, and then most importantly, also ensuring that there is a highly skilled team at Arthex to ensure maximum outcome for these development programs. So, as usual, we want to hear from all of you live. Please submit questions. I have also curated a whole lot of questions, and after Arthex presentation, we are going to hopefully have a free-flowing conversation where we are able to answer most of these questions. So, without further ado, welcome Arthex, and we are going to hand it over now to the Arthex team for their hopefully super exciting presentation.
Thank you very much, Andy, for that kind introduction. And I want to thank the work you do and for allowing us to speak with the community. And thank you to all those of you who are on this call who either are living with myotonic dystrophy or have a loved one with myotonic dystrophy or are here out of interest in this disease, which has multiple effects. We are really very honored to be able to speak with you today. So I'm Judy. I'm Chief Medical Officer, as Andy said, and I'm going to kick us off, and then Beatrice will speak, and then I'll come back and talk to you a little bit about our clinical trial. Next slide, please. So Arthex is a relatively new company. Andy said we were founded in 2019. Years, we have progressed an incredible amount thanks to the leadership of Beatrice, and we are now in the clinic. So to go from having a concept in a university setting to being in the clinic is quite remarkable. And what we're doing as a company is trying to build a company that focuses on rare and genetic diseases. Our first disease that we are looking at is DM1. I will also tell you towards the end of my presentation what we're doing in other forms of myotonic dystrophy. But these are important diseases with a lot of unmet medical needs, and there's no need for me to tell you that. You know it only too well. One of the and that we're very hopeful for the community is that we have a dual mechanism of action with our lead compound. So you're going to hear about our lead compound, ATX-01, and Beatrice is going to walk you through how we think it works or how it might work in DM1 based on some of the animal data and other studies her group has done. But the interesting thing is that we have a dual mechanism of action, and she'll explain that in detail. And we also have built our company so that we can then expand into other diseases. And again, we're going to talk about DM2, we're going to talk about congenital DM, but also other opportunities. So that's just a quick introduction. And if you go to the next slide, which is going to recap how we've approached myotonic dystrophy type 1. And one of the things, I mean, I don't need to tell this audience how DM1 affects patients and how it affects them not only throughout the course of their life, but how it affects so many different systems. So we talk about myotonic dystrophy being one of the muscular dystrophies, which of course it is, and it has prominent effects in the muscles, including the weakness and the wasting of the muscles, and of course the myotonia, but it also affects virtually every other organ system. And so it's important when we develop drugs for myotonic dystrophy that, yes, we deal with the muscle aspects, but that we also address the other systems that are affected, such as the central nervous system, where we see sleepiness and we can see some other disturbances of thinking and memory, the cardiovascular system, where we see weakening of the heart muscle function and irregularities of the heart rhythm, the respiratory system, even the vision can be affected with cataracts, the endocrine system can be affected with diabetes and other endocrine disorders. GI tract abnormalities are often overlooked, but I know from speaking with patients with DM1 how considerable they can be and what an effect they have on the patient's day-to-day life, et cetera. So we need to be able to address not just the muscle symptoms, but the whole panoply of symptoms. And we know that, there are different forms of DM1. There's a congenital, childhood, adult, and late onset forms. And ideally, when we are developing a drug, we need to address all of those forms. Of course, we are focusing, and I'll come to that when I talk about our clinical trial, on adult DM1 first, but it doesn't mean we're ignoring the other forms. And although DM1 is considered a rare disease, it's actually a common rare disease. It's not as rare as some other diseases, and so it should be getting a lot of attention. And I think indeed it is, as Andy showed you with his list of his outline of drug development in this condition. There's the incidence of the genetic fault might be as high as 1 in every 2,100 births, which is really quite significant. And we know, unfortunately, that it is underdiagnosed clinically, and that there are probably more patients that have the condition and can pass it on to their offspring than we are aware of. So without further ado, we're going to talk a little bit about how we are approaching DM1 with our lead compound, ATX-01, and I'm going to hand over to Beatrice, who's going to talk to you about how it might work. Beatrice?
Thank you, Judy, and thank you, Andy, for the introduction. You did it perfectly well by explaining, so you make my work easier. As you all know, the genetic cause of myotonic dystrophy is quite well known. It is originated in the mutation in the DMPK gene. This mutation is the expansion of the CTG triplet in a non-coding region of the genome, which is generating this toxic RNA. And it's called toxic because it is retaining MBNL proteins. That's the classical description of the mechanism of disease. This retention of MBNL protein is producing aberrant splicing, so the protein can no longer perform its function. It gets accumulated in the nuclei, forming these dots that you can see in the photo on the left. These are the ribonuclear foci when MBNL is sequestered. But we have described that there is a second mechanism that is producing further loss of function of the protein. We have described that miR23B is upregulated in the muscle of the patients. And this microRNA is blocking the production, the translation of MBNL proteins. So these two mechanisms are both contributing to the final loss of function of MBNL, which is producing the symptoms in the patients. As Andy has described perfectly well, most of the companies are focused on the approach of DMPK, either depletion of the toxic RNA or blocking the interaction between the protein and the RNA. But we believe that MBNL has the main role on the symptoms and it deserves further attention. And in fact, if you go to the next slide, we were quite happy to see recent proof of concept in human data already. This was yet data generated by our colleagues of Avidity. We have borrowed this slide when they have described that the functional rescue they have seen in the patients correlates very well with the levels of free MBNL available to perform its function. They have not seen this correlation with DMPK levels, only with MBNL. So that's why we believe that our mechanism of action can have the strongest effect on strongest therapeutic effect, because our molecule has, as Judy described, a dual mechanism of action, and we are affecting the two mechanisms that are producing the loss of function of the protein. First of all, we have seen that we produce a destabilization of the toxic DMPK foci. So we reduce the level of DMPK and therefore we are releasing MBNL. But in addition, we have a direct mechanism by blocking the miR23B inhibitor, and we produce an overexpression of MBNL. So at the end, adding these two mechanisms, we will have further levels of MBNL available. If you go to the next slide, I can show you a little bit more about how is our molecule, what is ATX-01, because our final molecule is a combination of two parts. One is the AntiMIR oligo. This is an RNA sequence that is blocking microRNA. So it's the sequence and the chemical modifications that we have done to this RNA were specifically designed for having high specificity for this target and also having low potential of targets. Also to improve potency, increase durability and affinity. And all the chemical modifications we have included in these molecules were already used in other drugs that have already been approved and used in humans. So we believe that the chemistry is safe. In addition, we are combining this AntiMIR with a hydrophobic conjugate, that is oleic acid, and is improving the delivery of this molecule to the right tissues. So with this combination, what we have done is to improve the amount of drug in tissues affected by the disease, such as mainly muscle and nervous system. We know that we have higher target engagement in the muscle. We have seen this in vivo already in the different models of the disease that I will show you now in next slides. We have seen also that still come back. Yeah, thank you. We have also seen that we can go through the BBB, through the blood-brain barrier. So by an intravenous injection, we could have effects in nervous system. We are increasing the half-life of the molecule in the system, in the plasma, and also in cerebrospinal fluid. So we have a longer duration of the effects, and it has a chemistry which is also safe because it's a natural product. So the final combination of these two components is producing a highly efficacious molecule, specifically in muscle and nervous system, with a wide therapeutic window that allow us to use it in ascending doses in humans. Okay, yes. Now go to the next one, please. This is just a short summary of how do we have this, or how do we see this dual mechanism of action in human cells. As an example, here we have used human DM1 myotubes from biopsies in the patients. We have one control which is not treated, and cells which are treated with ATX-01. And here we are quantifying DMPK expression and also MBNL levels. So as you can see, with our drug ATX-01, we are decreasing importantly the levels of DMPK and increasing MBNL. Please go to the next one. What I've shown you previously is in vitro studies, but we have also done a lot of work in different murine models. So the first one is the HSALR generated in the laboratory of Charles Thornton in the University of Rochester, is the best described model of what will be adult onset DM1. These mice are expressing the mutation only in the skeletal muscle, so they only have muscular phenotype. And here what we were doing was to inject once intravenously, one injection every 2 weeks, and then analyze the molecular biomarkers in the muscles, quadriceps or gastrocnemius muscles of the animals 2 weeks after the last injection. And during all the period of the study, we were also analyzing the grip strength of the animals. That is what you can see below as functional rescue. If you see these animals normally has a reduced strength. You can see the disease animals are in the gray line, while the wild type strain is the purple rectangle, is the FBB strain. But if you see with treatment of ATX-01 at different in a range of concentrations between 6 and 24, even after the first injection with the lowest dose, we already have a complete rescue of strength in this animal that is endurable during all the period of the study. In the muscles of the animals, we have quantified, of course, MBNL levels and also splicing rescue. So what you see here as molecular rescue, overall PSR means percentage of splicing rescue. And what you see here in every red dot is a composite of a group of splicing events that are regulated by MBNL. So as you can see, with higher doses of the treatment, we are reaching the level of the wild type condition. In addition to rescue splicings, we can see in the right panel the effects of on MBNL levels. And as you can see, we see a nice dose response, dose upregulation of MBNL in the muscles, but not only in skeletal muscle. We have also seen it in heart, also in diaphragm, which is a very important muscle for the respiratory problems of the patients. And surprisingly, we also saw upregulation of MBNL in the brain, in different regions of the brain, in fact. But we didn't know if this was something direct, because the therapy was really going into the brain, or if it was just indirect due to the effect on the muscle. But it was a very, very surprising observation. On the contrary, in the liver and the kidney, we only see upregulation of the target of MBNL at the highest concentration. So it seems that we have seen a preferential MBNL elevation in the tissues that are more affected by the disease. Given the results we have seen in the brain, we decided to move to a different model. In the next slide, you can see the results we obtained with the DMSXL model generated in the laboratory of Genevieve Gordon. We performed these studies in collaboration with them. This model is expressing a longer track of repeats, because it's above 1,000 repeats in all the tissues. So the problem is that we are using here adults, and in the case of the adults, they are expressing very small amount of toxic DMPK in the muscles, but a nice amount of toxic RNA in the brain. So they have mainly cognitive dysfunction, but very mild muscular dysfunction. So this is the right model to evaluate the potential effects we could have on the cognitive DM1 alterations. Here we were injecting 4 times, where we performed 4 intravenous injections, 1 injection every 2 weeks, and again analyzed the data 2 weeks after the last injection. In different regions of the brain, but I show you here the results we obtain in the temporal cortex, because it's the part of the brain which is most importantly affected in the patients. And as you can see, in these animals, the drug could improve MBNL levels and also MBNL2 levels in this region of the brain, and also reduce DMPK expression. And as in global, we were also increasing the percentage of splicing rescue. So we were able to rescue more than 30% of the splicing in these animals. And the better thing is that in addition to see molecular effects, we know that these molecular effects are meaningful because we have seen a behavioral rescue also in the patient, in the animals. So these animals with the disease that you can see in the light purple bar, when you put them in the center of an open field, they tend to go, a normal animal will tend to hide into the corner, but these disease animals, they stay in the center. They can rest in the center and spend a lot of time there. So, and this is not normal in a mouse. So what we have seen is that with our treatment in the dark purple bar, we are completely rescuing these exploratory behavioral abnormalities that have been reported in this animal. So that means that we are reaching the brain with an amount of drug which is enough to produce a functional improvement, at least in the animals. If you go to the next slide. After all this proof of concept of efficacy, of course, we moved into the toxicology studies just to make sure that the drug was safe to start the studies in patients. As a summary, all the IND-enabling studies we have performed have been quite positive or quite clean, let's say. ATX-01 is remarkably safe and well tolerated. We haven't seen any genotoxicity in vitro nor in vivo. In the studies with non-human primates, we haven't seen any immunotoxicity. And we have seen that we have a therapeutic window which is wide enough to support ascending doses in human patients. One important thing is that the starting dose we will be using in humans will be the equivalent to the a dose that was therapeutic, at least in the animal models. Of course, the translation from mice to human, you never know what can happen, but at least we suspect that we might be in therapeutic doses, hopefully. And with all these studies, we have complete all the preclinical program for initiating this phase 1-A that now Judy will give you more details about this study.
Thanks, Beatriz. Okay, great. So let me tell you about our first in human study. But before I do, I just want to recap what Beatriz said. So all the work she and her team have done have shown us that we have a drug that has biological effects on cells from humans with DM1 that has effect in 2 animal models. And these effects are in reducing toxic DMPK RNA and increasing MBNL and rescuing abnormal splicing and leading to functional effects in the animals, whether it be increased muscle strength, grip strength in the mouse, the HSA-LR mouse. And also what we didn't show you was improvement in myotonia in the HSA-LR mouse, or improving exploratory behavior in the DMS-XL mouse. So we have very positive results from all the animal and preclinical work, plus a very nice, clean toxicology profile. And we looked at the drug. We looked at the safety of the drug in mice, rats, mini pigs, and monkeys. And so the next step, of course, is then we felt comfortable starting a first in human study. So this study is called the ArthemiR study, and we have started this study, we're delighted to say. It is a first-in-human study of our drug ATX-01, and we are comparing ATX-01 to placebo in the study. So some patients are going to get the active drug, and some patients are going to get a drug that looks like the active drug but doesn't contain any actual drug in it, called a placebo. Now, we say the study is double blind because the patients who are participating in the study won't know if they were getting the active or the placebo. Neither will the doctors or nurses or pharmacists who are looking after them, and neither will we at Arthex or our partners that we're working with. The only time we will know is at the end of the study when we do what's called unblinding and we look at the code and see who got what. And this is the best way to do it, to make sure that we are being scrupulous and that we are actually seeing real effects. So double-blind, placebo-controlled study. And we are doing, in fact, the study has two parts, you could say two studies, what we call a single ascending dose study and then a multiple ascending dose study. So we've got two parts that are almost two separate studies, and I'll explain those in a minute. And in this very first study, we're looking at patients with classic myotonic dystrophy type 1, the adult form. And based on our mechanism of action, we do think the drug could work in other forms. But when you are doing a first clinical trial, it's important to make sure that you have a group of patients that are homogeneous, that are all like each other, and that you start with a restricted group. So we've chosen to start with the adults. Doesn't mean we're not going to look at other groups down the road, but that's how we're starting. Now, the objectives of this study, as this is a first-in-human study, our first goal is to make sure that ATX-01 is safe and well tolerated by patients. As Beatriz said, the animal data look good, but humans are not mice. They're not even monkeys. So even though we've got nice data in the animals, you just don't know until you go into humans. So that's our primary goal. And because that's our primary goal, we are starting with very low doses and gradually increasing them. However, in addition to looking at safety and tolerability, we think we can also assess how well this drug might work in terms of benefit for patients. And we're going to look at that in two ways. We're going to look at some clinical outcome measures that you may be familiar with, things like looking at myotonia by measuring video hand opening time, looking at time to walk 6 meters, time to stand up from a sitting position, etc. And we are also going to do muscle biopsies to look at what we call target engagement. Target engagement means that when you look at the muscle, do you see the increase of MBNL protein that we should see, and that we saw in the animals? Do we see the improvement of splicing index that we saw in the animals? So we have a lot of objectives with this study, and we are really counting on the DM1 community and physicians and other healthcare staff to help us make the study a success, because we can't do it alone. Next slide, please. So here is the overall design. As I said, there are two parts. On the left, in the teal color, you can see the SAD part and then the MAD part. Now, on this slide, they look separate, but in reality, they will be overlapping. So in the SAD study, we are going to have 4 cohorts, by which we mean 4 different doses are going to be tested. So we're starting with a dose of 16 milligrams in cohort 1A. And if all goes well, we are going to go to cohort 1B, and the dose will be doubled. It'll be 32 milligrams. And if all goes well in that cohort, we're going to go to cohort 2, which is going to be 64 milligrams. And then to cohort 3, 128. Now, going from one cohort to another is going to be determined by what we call a Data Safety Monitoring Board, or DSMB. And this is an independent group of scientists, people who are experts in DM1, statisticians, and people who are experts in kidney disease, immune reactions, and so on. And they will look at the results. They will be unblinded, look at the results, and they will be able to tell, look, this is safe to continue, or no, we need to we need to pause and take it, reassess. They may decide, look, we've finished the cohort with 8 patients, but we'd like to see a few more before we move to the new cohort. They may say, we don't want to double the dose fully, we want to increase it only slightly. So the DSMB is going to play a critical role. They're independent to make sure that we're at all times protecting the safety of participants in our study. Each cohort is planned to have 8 participants. As I said, that can vary. Those 8 participants, 6 will be assigned randomly to active ATX-01, 2 will be assigned randomly to placebo. And as I said earlier, no one will know who gets what. And they will get a single intravenous dose, and they will be followed up for 12 weeks. And we'll talk about that in a minute. The MAD study is very similar to the SAD, except for the fact that each subject will get 3 doses. So they're going to get a dose at the start of the study. 2 weeks later, they're going to get a second dose. And then 2 weeks after that, they're going to get a third dose. And then they're going to be followed for 12 weeks with various assessments. We are starting the dosing in the MAD at the 32 milligram level. Now, just to go back to what Beatriz showed you, in the SAD, I said we were starting at 16 milligrams. But as she said, even at that low dose of 16 milligrams, if you look at the equivalent dose in mouse, we saw some beneficial effects. So even at our starting dose, we might see some effects in humans. But we think overall, by the time we get to cohort 3, we should be in a good therapeutic dose, but we don't know. And at the end, the patients who enroll in the SAD, if all goes well, and if they want to continue, we will invite them to enroll into the MAD, the multiple ascending dose. At the patients who have completed the multiple ascending dose, the MAD study, will be offered a long-term follow-up study. We are hoping to be able to provide drug to all patients who participate in the study. We can't do that right now because we don't have the animal toxicology studies to support giving long-term dosing, but we hope to have that soon. And then we can give everyone who's completed the study some ATX-01, assuming that all goes well. Next slide, please. So who can enroll in the study? Well, I hinted at this earlier. We want to have a fairly restricted group because if we have a group that's too varied, it becomes very difficult to see if your treatment is having any effect. So we have to narrow the who gets treatment. And we do this with what we call inclusion and exclusion criteria. These will be similar to some of the other studies, I believe. So we're looking at adults, they could be men or women, who have a diagnosis of DM1 and who have more than 150 CTG repeats. So if you don't know how many CTG repeats you have, we will do the test and let you know. But we want patients who have at least 150. We want them to be ambulatory, meaning that they can walk. So if you're not allowed to use any aids or devices to walk other than an ankle foot orthosis. And we want patients who have myotonia and have defined as more than 3 seconds on the test called a video hand opening time. And of course, the patients have to be fully informed and sign informed consent forms. We are excluding patients with congenital DM. We're only looking at those who have an onset of DM at an older age. And we are doing muscle biopsy. So if there's any reason why you can't have a muscle biopsy, say you have a bleeding disorder, then you can't enroll. We want patients who are not on anti-myotonia medication. So if you're on anti-myotonia medication and can't stop it, then you wouldn't be eligible. But if you can stop it, you can wash out from your anti-myotonia medication and participate. And we are because this is a first in human study, we're being fairly strict on making sure we don't have patients with advanced cardiac disease in the study. We have not seen any problems with our drug from the cardiac perspective in the animal, but we want to be cautious. Next slide. And I said we are going to follow the patients over the course of 12 weeks after the single dose in the SAD part or after the last dose in the MAD part. And how are we going to follow the patients? Well, we're going to do safety and tolerability assessments. Every time the patients are seen, they're going to be asked about any adverse events that have occurred. We're going to do blood and urine testing to look at various laboratory parameters. We're going to do electrocardiograms and Holter, which is prolonged electrocardiogram recordings. We are going to do what's called pharmacokinetics, and this is where we take samples of blood and urine and measure the level of ATX-01 in the blood and urine to see if it's getting in and how high the levels go. And if we have enough muscle sample tissue from the muscle biopsy, we're going to look at the levels of ATX-01 in the muscle. We are also going to do what's called pharmacodynamics. And this is what when we look at the muscle biopsy samples and we measure the MBNL protein levels and we measure the splicing, just like we did in the mice, we'll do this in the human and see, are we in fact doing what we said we would do, increasing MBNL protein and improving splicing. And then in terms of clinical outcomes, these are things that are not in a laboratory, but that are live. We're going to do a whole host of muscle and function testing. So we're going to see if there's any improvement or not in myotonia. We're going to do a 10-meter walk-run test. We're going to do a timed up and go, and some of these tests that you might be getting anyway in the clinic when you see your doctor. We're going to do a measure of lung function, the forced vital capacity, where you blow into a tube. So these are things that are quite commonly done in these types of studies. We are also going to ask your healthcare practitioners to complete some scales. We're going to ask them how they think you're doing. We're also going to ask you how you think you're doing as a patient, but we want to get the healthcare professional's opinion. And we're doing something called an apathy evaluation scale, just to see if there's any effect on the level of interest that patients show to various activities. And then, of course, it's really important to get information directly from the patients who are participating in the study, and we do that through patient-reported outcomes. Fatigue and sleepiness is a big issue in this disease, so we're going to use a fatigue and daytime sleepiness scale. We're going to use a scale that looks at activities of daily living called DM1 Active, and we've developed our own scale that we're going to test for the first time in this study, which is called the impact of symptoms on activities of daily living. We're going to ask you how you think you are doing with the Patient Global Impression Scale. And we're also going to look at whether you are having any gastrointestinal symptoms and how they fare over the course of the study. So it's quite intensive, but we want to try to extract as much information as possible. And again, coming back to one of the earlier slides, this is a multisystemic disease, and we have to test everything, things in the lab and things in the clinic, but not only just muscle, but all the different systems. Next slide, please. So we filed our IND last year, and we have the study approved now, first in the US. We also have approval to run the study in Canada, in Europe, and in the UK. And we had our first patient enrolled in July, and we are slowly getting different sites set up and ready to start enrolling. So we now have 2 sites in the United States that are open to enroll patients, a site in Italy and a site in France. And we're hoping over the next few weeks to be opening up the remaining sites. We have 9 sites that are definitely participating in this study. We'll see if we add any more. I would love to have sites everywhere, but we can't for this first study, but it doesn't mean we won't for the subsequent studies. Next slide, please. And as I said, and we're focusing on DM1, but we have very strong reasons to believe that our drug will work in congenital DM1 and also DM2. And we are also looking at some other muscular disorders with completely different oligonucleotides. If you go to the next slide, I want to talk a little bit about DM2 because we always get questions about DM2. And although DM2 is similar to DM1 in the fact that you have the sequestration of MBNL proteins, the trapping of MBNL proteins in the nucleus, it's a completely different gene. And it's a gene, the CNBP gene. And what happens in the CNBP gene is that instead of triplet repeats, there are quadruplet repeats. But the same thing happens. You get these hairpin loops and they trap the MBNL protein. So MBNL protein is not available to do what it normally does. Now, the disease is also multisystemic, but has a different clinical picture than DM1. You do see some muscle impairment, but you also see a fair bit of pain, and you see a very different you see some CNS, but it's different. And there's quite a high incidence of cataracts, and you can see some of the other symptoms that are seen. The muscle defects within type 2 are also very different, whereas in type 1, it tends to be ankles and wrists and fingers. In DM1, it's what we call distal muscles, hands and feet. In DM2, it's what we call proximal muscle problems. It's more the hips and the shoulders. It is thought to be rarer. DM2 is thought to be rarer than DM1, although that varies by region. And we know there's some countries where DM2 is more common than DM1. It tends to start in adults. We don't rarely see children with DM2, and it's but again, it comes down to the sequestration or trapping of the MBNL proteins. Next slide. So very little is being done in DM2 in the clinic. In fact, there's nothing that I'm aware of in the clinic at the moment, but people have tried to approach DM2, but there's a concern that if you try to block the CNBP gene, the toxic mRNA, that it might lead to muscle wasting. So that approach of blocking of destroying the toxic RNA that works in DM1 doesn't seem to be going to be useful in DM2. On the other hand, if you go to the next slide, in DM2, we think that approaches that look at trying to release or upregulate MBNL will be viable in DM2. And of course, that fits very well with what our agent, the same drug that we're using in DM1, could also work in DM2. And there's some papers here, we don't have time to go through them, but they sort of support that concept that perhaps this could work. Now, the difficulty with DM2 is that there aren't very good animal models. So Beatrice and her team are currently working on getting cells from patients with DM2 and doing studies on those cells with ATX-01. Next slide, please. So just to round off and get to the questions, we firmly believe that we are at a new turning point for the development of agents for myotonic dystrophy. We're really, really excited about the potential of ATX-01. We're excited about the whole progress in the field of DM1. I think it's absolutely amazing, and we are so grateful to be part of this. And again, we thank the Myotonic Dystrophy Foundation. We thank our supporters. We've had a great support from various governmental agencies in Europe and in Spain. We thank, and most of all, we thank the DM community for partnering with us and leading us to improved outcomes for all patients with all forms of myotonic dystrophy. Thank you.
Thank you so much, Beatrice and Judy, for this very detailed and in-depth presentation and all the evidence you presented today. So before we jump into the Q&A session again, one more time, there's an opportunity to ask questions. Submit them. You can send them anonymously, but please do, because then we'll have a robust conversation. So with this, I think we'll go now to the Q&A part, and I want to start with, so I grouped the questions based on clinical trial practice, mechanisms. But before we go there, I want to pick up on the DM2 story. We often hear, right, that DM2 is the neglected form of myotonic dystrophy, and it would be great if you could speculate a little bit when you think, right, when you feel that your compound or might be ready for prime time and enter the clinic for DM2 as well.
Yeah, you don't ask easy questions, do you, Andy? That's always going to be a tricky one. Well, let me tell you what I think. Beatrice, you can jump in. I think the first thing we need to do is make sure that ATX-01 is safe to administer to humans and that it's well tolerated. So we need to do the ArthemiR study. I would also like to see that it works in DM1, because if it doesn't work in DM1, we really have to think about, well, why would we think it would work in DM2 if it doesn't work in DM1? So I would like to complete that study. The nice thing is that we don't need to do a whole new toxicology program for DM2 because we have the toxicology program, so that saves us some time. But I would also like to see the results of the studies Beatrice is working on right now with the cells, the in vitro cell studies, because I'd like to have some evidence that we have a biological effect in cells. There is unfortunately no animal model, so we can't, we're speaking with groups who are developing them, but I think if we have to wait for an animal model, it might be too long. The other difficulty that we have, and this is where I want to reach out to the community, and I know you'll support this, is that we don't have a lot of natural history data in DM2. So as compared to DM1, DM1, we do have a lot of natural history data, and designing a study, it's much, much easier when you have natural history data. And I know there are efforts from various groups and some people like in Germany and Serbia, they are collecting natural history data. I know in the US, I know that the MDF has collected data from some from DM2 patients. There are ongoing efforts to collect data over time. And I would urge anybody with DM2 to participate if you're asked to participate in these studies, even if there's no drug involved. It's so important to us to get these data so that we can design better studies. And in particular, know what to measure in the studies, because in a DM1 study, we can measure things like ankle dorsiflexion, but in DM2, ankle dorsiflexion isn't affected. It's a different muscle groups that are affected. So we have to have measures, and we don't know what the best measure is. So we need the natural history data. Beatrice, do you want to add to that?
Yeah, no, as you said, something that has also limited the development of DM2 drugs is the lack of phenotypes in the models in vitro and also in vivo. There is no very mild splicing defects. We are working with the cells we find available, and I know that also in the laboratory of Laura Ranum, they are developing a DM2 mice that still needs some work on it, but it might be a good way of testing first in the animals before going into humans.
Well, you know that you have, I'm very glad you mentioned the two things. So natural history study, we are your greatest supporters and allies there, and advocates for even longer term studies than, for example, what we have in DM1. And obviously we need the same for DM2. And then patient involvement, right? The patient voice is extremely important, so we know what we need to measure. So great. So now let's go to the clinical trial design. We always get questions. How do you decide on your trial sites? Why don't we have sites in Scotland or in Germany or in, you name it, whatever part of the world? Maybe if you could quickly summarize what your criteria are for those, and how you go about selecting more sites, right, as clinical trials show positive outcomes.
Yeah. That's a really great question. So the way we started out is that we where is the money? We go where the money is. Where are the patients? So we know there are different groups that have databases of patients and that have been following patients, and most of the sites that are involved in our study are sites that have participated in the Myotonic Dystrophy Clinical Research Network natural history study, because we know that they have the patients, they've been following them, they have a database of patients, so it's much easier to make sure that we have the patients to enroll in the study. We are also working with the DMCRM. This is this network of sites, because there are a lot of different companies that are doing studies in DM1, and we want to make sure that we're not stepping on each other's toes. So what we've done by working with the network is that some sites are doing multiple studies, but we want to try to avoid that, so that some studies are done at some sites and other studies are done at other sites. So there's that. And then because our first in human study is relatively small, we only need about 9 sites. We might be wrong, we might need more, but at the moment we're planning on 9 sites, but we are definitely going to need more sites for the subsequent study. And then we will plan to expand and try to cover a broader geographical region.
Thank you. So let's go into clinical endpoints, right? You have essentially effects on muscle function, but you also have profound, at least in animals, profound effects on the CNS. Can you speculate what the community needs to do to advance knowledge in terms of these CNS phenotypes, right? So we know actually that a drug like yours is potentially very active, right? Combating daytime sleepiness and all of these associated clinical manifestations.
So a really important question, and one that we have struggled with and continue to struggle with. So we have limited CNS assessments in the ArthemiR study, and the reason for that is that it's a small study, and usually to see effects on the CNS, you need big studies and you need to follow patients for a long time. So we don't think in this particular study that we're going to see a lot of change on CNS function. The other thing is that we are selecting patients for the ArthemiR study who have muscle disease, and sometimes if you select a population that has muscle disease, they may have less CNS disease. So if you really want to study the effect of your drug on the CNS, you really have to select for a population that has CNS disease. So I think that what we're going to have to do, if we really want to explore this, is look at patients who have CNS disease. And we know, again, childhood and congenital DM have more CNS disease, and that might be the better way to assess that, is to do a study in those conditions. So that's why we've kind of gone halfway. We have some CNS assessments in the ArthemiR study, but not a whole lot.
Great. I know we have to be respectful of time because you have another engagement. So maybe one last question with regards to trial design. So we are dealing with a slowly progressing neuromuscular disease, and the trial duration is 12 weeks. Do you think that is sufficient? And what does the community have to do in terms of achieving detecting maximum benefits, potential benefits of the drugs?
Yeah. So do I think 12 weeks is sufficient to get a drug to market? No, I don't think. No one would believe that. I think the purpose because it's a first in human study, we are being a bit modest in our ambitions here. And we are wanting just to see, well, first of all, as I said, safety. Is it safe? And again, you don't have the full safety picture until a drug has been on the market for years, but you doing a first in human study, a phase 1 study, can give you an idea, is there anything major that would happen immediately? So that's the first goal of the study. And also, when we originally designed this study, we thought, well, we should be able to see something in terms of what we call target engagement, looking at the MBNL levels and the splicing in the muscle. We might be able to see that in a short study, but we didn't think originally that we would see anything on the clinical outcomes. But the results from the avidity studies and the dying study have shown that even within short periods of time, you can shift things with effective treatment. So now we're actually thinking more positively. So I think we might see something. Does that mean that the effects are going to last? That it's, no, it doesn't. We would need to do more definitive, longer term studies eventually. But I think as a first step to see whether there's something there to follow up on, I think this type of design makes sense.
Well, thank you for this very optimistic, right, response. I think so. Judy is in Canada. She has another engagement, and Beatriz is in Spain, and she needs to go to bed. So I think what we're going to do here, let's end here. Thank you very much again to the two of you for your insight presentations, and we will have you back for sure, because we all want to hear how the study progresses. So if today's program liked you and you liked what you saw, please consider showing your appreciation with a donation to MDF. Judy kind of highlighted it. If you are living with a disease, please participate in our registry. That's important, especially going forward, and we need to decipher between or distinguish between CNS and, for example, muscular phenotype. With this, I want to end. I want to thank you again. Big thank yous also for the community. We had some thank yous in the Q&A sessions, and thank you for your time. And until next time, thank you very much.
Thanks to you all for your attention.